Okay, before we start this life cycle of a DNA virus, I want to throw to you guys a simple question here. How many of you like baking or cooking? Okay, so but the problem here, let's say your house don't have kitchen, your house don't have the oven for you to cook or to bake. Okay, you don't have the utensil for you guys to use. What you guys do? Yeah, what you guys will do? You go to borrow the kitchens, right? You go to rent the kitchens, you go to borrow the kitchens. Okay, you go to the next door and you ring the bell. Hello, may I borrow your kitchens? Okay, so what you bring to that, because you don't have utensil, you have nothing, Karana. You don't have the utensil, you don't have the baker, you don't have the mixer. If you go talk about baking, then but you have a recipe, right? You have the recipe. So you basically is like a virus. So this virus, they can't produce their own, or reproduce. They can't. Why? Because they only have the cookbook. They don't have ingredient. They don't have the machine. They don't have the ovens. They don't have the cooking utensil. So definitely they cannot reproduce outside the host cells or infected cells. So understand the concept now. What virus do? Virus are going to borrow. When you use a nice term, borrow. If I use the words, the more appropriate word, basically hijack. Okay, so what you need to do now, because we know that you only have, the virus only have the cookbook, the recipe, but they don't have the ovens, they don't have the mixer. So they go and borrow. Borrow from who? Borrow from the host cells. So what they do? So they go into the host house, they go to, like us, go to the neighbor house, and then we start to use all those machines already. Okay, we use the mixer and we blah, 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 use already. Oh, not enough ingredient, no ingredient, flour, don't have. Take from the neighbor, the kitchens. Okay, steal it, right? We steal from who? We steal it, okay, we check for the flour, we check for what? Butter, we get the, for example, sugar, and so on. Okay, we steal. So same, this virus also have the same thing. They only have a recipe. They don't have the ingredient. They steal from ourselves. Are you clear? They steal from ourselves. Okay, but something better we compare to the virus. For us, we finish using, we have to clean. Okay, we have to clean, put back the thing, and then say thank you. Okay, right? We our cake that we bake, maybe we share half with our neighbor, right? But virus won't. Virus is more nasty. In this case, I use the word. Why? Virus, when they finish everything, they won't help us to clean. What they do? If I want to analog, I use the same word. Basically, you finish using your neighbor kitchen already, you destroy the kitchen. You finish using the kitchen, you destroy the kitchen. This is what virus normally do. So virus reproduce inside the host cell. When after they finish reproduction, what virus normally do? They lyse the host cell. They're going to bomb the host cells, to kill the host cells. Therefore, we say that the virus actually is a parasite. Okay, they enter into our host cells, into our cells. What they do? They use our cells ingredients, use our cells machinery to build more viruses. And at the end, they lyse our cells and then they kill our cells. So when the cell destroy already, then it leads to the disease. Like for example, HIV destroy our immune cells. When the HIV destroy our immune cells, means what? They are going to cause the immune system, or we call immunodeficiency disorder, or we call it the AIDS, acquired immunodeficiency disorder. Are you clear? So this is slightly different between us and also the DNA virus. Okay, so look at the process here. You look at the process. So few terms that I'm going to introduce. Okay, learn whatever you can, but do not need to memorize it. Okay, no question will be asked at this moment. Okay, so first you look at this. As I said, that all viruses, they must have the capsid and they must have the DNA. Correct enough. Okay, so what actually make them, the virus, know which host cell to attack or to infect? Basically, they do have a what we call the interaction. So the virus must have a form of interaction that actually allows the host cell to take in the virus. Can you see that? So what is the mechanism here? The mechanism involved here is the receptor. Now, what's mean receptor? You try to imagine why, okay, a very, very simple idea here. Okay, very, very simple idea here. Why virus, particularly we talk about COVID nowadays, okay, the coronavirus. Why COVID didn't attack our skin cell? Luckily, correct or not? If let's say COVID attack our skin cell, then you don't need to go out of the house anymore. You have to wear the protective suit, cover all your skin, cover everything, right? But why this virus attack our lung cells, the COVID? Basically, it's something like the welcoming signal here. Come and attack me. Okay, so this welcoming signal or welcoming protein is called as receptor. Okay, to receive. So that's why receptor. Can you see that? So something on this, okay, on the protein code here allow the interaction between the virus and also the host cell. Are you clear? Okay, so it's something like our host cell say, yes, please come and attack me. But in fact, it's not like this. Because why? The host cell won't want the virus to attack them. It's just that so coincidentally, they receive the virus, the wrong person. Are you clear? The wrong person. So in terms of COVID, okay, because nowadays everyone talk about COVID, in terms of COVID, that is the proteins on the capsid, which known as a spike protein, the S protein of the COVID, which can interact with the receptor of the host cell. Once they interact already, then the host cell can take them in. Are you clear? Okay, so the host cell take them in by a process called receptor-mediated endocytosis. Basically, open the gates, allow you to enter, allow the virus to enter. Okay. So with the viral DNA, as I said, that is a cookbook. Okay, it's a recipe. So we're using this recipe. What the virus can do here is, virus will use this. For example, if you want to bake a cake, you have the recipe. So now you start to make the mRNA. mRNA stands for messenger RNA. So this messenger RNA will be used to make the protein. So these proteins later will be assembled, and then you're going to lyse the cell, release more viruses. So when you release more viruses, the viruses are going to attack other living cells or other host cells. So the cycle will continue and continue. Okay, so a little bit complicated here. If you understand, then it's good. If not, then we're going to repeat this again in chapter 13, infectious diseases. Okay, so now I'm going to talk about this. I'm going to talk about this, how we can get the viral DNA to make into the protein in two-step work here. Okay, I believe you guys have gone through these two processes in brief, which is the transcriptions and translations. Okay, transcription and translations. Okay, now, what's it mean by transcription and translation? Okay, what's mean by transcription and translation? So first thing, you have the DNA. Okay, DNA contains the genes, which is the instruction. So our body have the mechanism to read the DNA. Okay, when you read it, then they form a message. So this message is coded in what we call the mRNA. Are you clear? So the process involved here is called transcription. Okay, basically copy. Transcript means copy. We copy the instructions from the DNA. Are you clear? So when virus infect the host cell, our body will help the virus to copy the information from the DNA. And this message is called mRNA. You don't expect that your cell have mouth, okay, have the voice box and tell, oh, please do this, do this, do this. Cannot. So how they communicate here, how they actually tell, okay, or send out the message is through this called mRNA by the process of transcription. Okay. So in a virus, when they have this mRNA already in the host cells, this mRNA will be translated. The process involved here is called translation. Okay, so by using translation, because mRNA is just a code, it likes a Morse code, right? It's something that, for example, you want to pass the message. Okay, you know that I won't be able to pass it to the person that I want to pass. I may have to go through, some intermediate person. Okay, but you don't want the intermediate person to know what you are talking about. So you use a Morse code. You use some, encrypted code. Okay, so this encrypted code, when passed to the intermediate person, when you look at it, I don't know what is the message here. Never mind, I pass to the final person. But the final person know how to read the code. Are you clear? So what is the final person? The final person here actually is our ribosome. So ribosome will have the ability to read this mRNA code and use this mRNA as an instruction to form polypeptide. Okay, so these polypeptides later will fold into protein or fold into the enzyme. Are you clear? So two-step work here. So it starts from the transcriptions. So based on the transcription process, it gets the mRNA. And from the mRNA, then will be translated into the polypeptide. So why viruses? They cannot reproduce by themselves. Why they need to hijack? Okay, why they need to hijack the host cell? Because of this. Host cells actually has a ribosome. You learn about this ribosome, right? Okay, so the host cells, they have a ribosome. It's a machine, as I say that. Ribosome, something like the oven. You don't have the oven, you go and borrow from your neighbor. So same goes to the virus. Virus, they don't have the ribosome. They don't have the machines to produce the viral protein, to produce the viral enzyme. They don't have the ribosome because they are non-living, non-cellular structure. So therefore, they have to steal it or hijack it from the host cells. So the host cell, very important component here, or organelles here, known as a ribosome. So ribosome help to translate the mRNA into the polypeptide. Okay, now in terms of the reproductions of the virus, involve two stages. Okay, even though this process takes place, right, from the DNA to get to the proteins, they happen in two stages. So the first stage is called early stage. Normally, early stage, they form enzyme. Enzyme for what? Enzyme for the DNA replication. Or we say viral DNA replication. Can you see that? Are you clear? Viral DNA replication. Okay. Also, after viral DNA replication take place, then we have the late process. Late process basically more to the protein. So once they already replicate enough already, then the virus need to resemble, okay? To assemble all this protein together. So for example, what kind of protein? Protein code or capsid. Are you clear? So two different stages. So first, early stage, produce the DNA, the enzyme that involved in the DNA replication. And also after finish, they continue with the late protein, which is involved in the protein code too. Later, we're going to assemble everything Can I see that? Okay, so it's similar to our baking. You like baking. So first thing what we do, we form the dough first, right? Can I see that? So, okay, we form the dough first. Okay, we mix everything together first, early stage. Late stage, what we do? We put it into the oven to bake it, right? So same goes to the virus. First, they produce enzyme first, mix everything. Then for DNA replication, produce every ingredients. Okay, then late we have the protein codes to assemble everything together to form new viruses. Okay. Okay, now so look at this life cycle of DNA virus. I have gone through with you guys a simple and basic one already. Okay, where we're going to learn it again in chapter 13. Okay, but for today, you can try yourself in these questions. Okay, so I'm going to explain some of the word here before you start. Okay, so first, adsorption. Now be careful the word at AD, not AB. AD, adsorption, basically means that interaction. Adsorption basically means interaction. Okay, assemble, assembly protein, no nothing. Capsid also known as the capsid also known as the protein coat. Then we do have endocytosis. Endocytosis is the process that take in the virus. Okay, anything that we take in to the cells, bulk take in. It's not like one by one take in. If one by one take in, we have what? Diffusion. We have osmosis. We have the active transport. Those one by one we take in into the cell. But when use the word endocytosis, means that we take in the particle into the cell in bulk. We don't break them one by one and take in. We take in huge quantity, large size. Okay. Genome means gene. The entire all the gene classified as a genome. Infection, no problem. Lyse means break. When you lyse something. Hemolysis, you learn hemolysis, right? Basically, it's the lysis or the breakdown of the red blood cells. Okay. Receptor, we learn about this. Transcribe is the work or is the process happen during the transcription, basically copy. Translate basically means that happen during a translation. We actually translate the mRNA into the polypeptide and proteins. Okay. So what you can do now is take 5 minutes. Try to answer the question here. Okay, so I stop the recording. Okay, so done for the answering. So I'm going to show you guys the answer. So during what? Okay, so during the infections, right? So this is an interaction. So you need to have the interaction first. So right. So the first answer during the adsorptions. Okay, during the adsorption. So the viral coat proteins or protein coats interact with a specific receptor proteins. Cannot, receptor protein. We have to actually interact with receptor protein. If not, they won't simply enter. Are you clear? So imagine the COVID. I said this coronavirus, the COVIDs. So they won't simply actually attack any other cells, right? So they only attack with those have the receptor that they can bind to or interact with on the surface of the exterior surface of the host cells. So the virus enter into the cells by this term called receptor-mediated endocytosis. Enter into the cells by endocytosis. Okay, and then the virus will travel to appropriate location. Right, you enter into the person's house already. You will find what? Find kitchens, right? So then in the cells and expose it. Okay. So next, the host cells. In this case, we want to transcribe the gene. Okay, the gene into the mRNA is by the process of transcription. So transcribe. So host cells enzyme transcribe the viral early gene into the mRNA. And subsequently, because we want to make the protein and the enzyme, right? So we by the process of translation. So translates, okay, this into the viral early proteins and enzyme, which replicates, okay, the viral DNA and induce the expressions of the late proteins, okay, by the late protein. So what are the late protein involved? It can be the capsid, also some assembly protein we want to assemble, okay. Can Okay, the enzyme that degrades the host cell's DNA, so supply the nucleotide for the synthesis of viral DNA. So the progeny virons are assembled in the host cells and released, okay, when the cells is lysed by the viral protein, and the newly liberated or freely, released viruses initiate another cycle of the infections in the host cells. Okay, a little bit of I forgot here. In other words, okay, sorry. Expose its genome. Okay, expose its genome. Okay, so do you get the whole idea? So do you need to remember this at this moment? No. Okay, at this moment, no need. But if you're interested to know, because nowadays we talk about COVID, you want to know more about how the virus actually replicates, basically almost the same process. Basically almost the same process. Okay, in general, how the virus, even though COVID, we talk about COVID is RNA virus. Okay, COVID is an RNA virus. So this one is the DNA, but basically the process, they are almost similar. Okay, so with this, I have done for today class. Okay,